Hi everyone, my name's Oscar Istas. I'm a senior here at UK studying biology and entomology, and for the past several years I've been doing research with Dr. Robin Cooper in the biology department. Our research started by taking a look at the disease septicemia, or sepsis. Some of you might have heard of this condition. It's a blood disorder that results from an immune response to a bacterial infection. Now normally your immune system responding to a bacterial infection would be a good thing, but in this case it's mistargeted and too strong. So rather than getting rid of the harmful bacteria like it's supposed to, our organs and tissues end up being affected, and they frequently shut down as a result. Now, the most common targets for septicemia are patients in hospitals who are already suffering from one illness. And in the United States alone, the death rate for this disease is near 30%. Now, the actual bacterial component that's responsible for bringing about this response is a small toxin located on the outside of some bacteria called lipopolysaccharides, or LPS. And while some research has been done into how LPS actually interacts with our cells and brings about this negative response, there are still a lot of unanswered questions. Now, in order to provide a few answers, our lab turned to a model organism, the fruit fly. Now, fruit flies not only have a very well understood physiology, having been involved in research for several years, but they also have a very similar nervous system to humans. You can see that they have a brain, central nerve cord, and segmented nerves that are all very similar to humans, which not only make them easy to understand, but it also allows us to draw some comparisons between the two species. The specific life stage that we looked at were the larvae, like this little guy. We would start by mixing our LPS toxin with cornmeal to create a contaminated food mixture. We would let our larvae feed on this for a few days, then transfer them to a clean plate and give them fresh food. We would then record how much of this food they ate to see if this toxin was impacting their digestion or their ability to feed at all. After this, we would transfer them to another plate and lightly tap them on the side, like you can see here, to see how they responded to that. This would tell us if their nervous system was being impacted by this toxin at all, and whether they were having any trouble responding to simple stimuli in their environment. So these two experiments, they would give us a good idea of whether these responses were similar at all to humans, at least on the outside. But the really important aspect of our research was looking at the cellular level. And to do this, we had to dissect our little larvae in such a way that we could see their nerves, like you can see here, and then insert small electrical probes into these nerves and stimulate them to get an electrical response. The electrical responses would show up as waves that we could see on a graph. So they were very easy to visualize and interpret. Now, the results of our research were surprisingly positive. We saw that larvae that had been exposed to the LPS consumed significantly more food when they were given fresh food than larvae that hadn't been exposed to LPS. That means that this toxin is doing something to influence their digestion and their ability to feed normally. Similarly, we saw that larvae impacted by this toxin responded to our light touches significantly less than those that hadn't been exposed to LPS. That means that their nervous system is being dampened somehow, and that this LPS is making it harder for them to function and interact with their environment, even on a basic level. So these two examples alone show us that there are a lot of similarities between how humans demonstrate their responses to this small toxin and how fruit flies do it. But what really excited us were our responses to our intracellular recordings. While our larvae were dissected and set on our microscope slide, we would look at the electrical activity in their nerves before and after we washed LPS onto them. So you can see down here that before we added the LPS, the electrical responses are consistent, similar, showing that their nerves are working completely normally. But as soon as we added the LPS, that decreased immediately. It became almost impossible to determine whether responses were there at all. This shows that the toxin is doing something to mask their nervous system, making it difficult for the cells there to function at all or to communicate with each other. And this is incredibly similar to what we see in humans in their inability to keep processing with their organs and tissues as part of the septic response. So looking at this, it might not have been a very positive experience for the larvae that we were experimenting on, but we ended up finding a model organism that's not only really well understood already, but very easy to obtain, that demonstrates a lot of similarities between humans. So looking at this, we hope to, in the future, not only use this organism as a way to understand the septic response better, but as a way to mitigate the threats that humans are still facing today, even in the United States. I want to thank you for your time and open myself up to any questions that you might have about my project.
Thank you very much. Great job. So, does sepsis affect the nerves or digestion in humans? Is that what you're looking for?
Great question. It definitely affects the nerves, as this toxin binds to different elements of our body, including nerves, that initiates a poor immune response, and our immune response attacks our nerves and other elements that these LPS toxins are binding to. So there's definitely a nervous component, which is one of the reasons that we thought fruit flies would be a great model organism for this. And some of the organs that end up shutting down as a result of this response are related to our digestive tract. And those are really visible symptoms from the outside. So we hoped that by trying to create a similar experience for these larvae, we could see if their responses were similar at all.
So ultimately, the goal of this step in the research is just to identify a model that can be used to research.
At the very beginning, yes. This toxin is relatively dangerous to work with, so obviously we can't do our experiments on humans or a lot of other organisms. So taking it from the bare minimum fruit fly is the first step to better understanding this toxin and how we can then compare that to humans.
Jump in. I know that a lot of different kinds of research have been done on fruit flies over the years, over the decades. If you were to take this type of research to another organism, what's kind of the next step? I assume that many other different kinds of research projects have started out with the fruit fly and then gone on to something next. What's next? The mouse, or are there other things that are sort of typical in that progression for examining something like this, the nervous activity of an organism?
Great question. So this research, after I completed doing this, other members of my lab already started tackling working with other model organisms at a very basic level. So the next step after this was working with crayfish, which have a slightly more advanced nervous system, but it's still very well understood, and a lot of labs even here on campus utilize them for nervous activity studies. So after we moved on from crayfish, we've started doing very minimal research on samples of human brain, actually. So we are definitely not at a point where we'll ever be able to test this with live humans, but we've gotten samples of important tissues and organs, and we're starting to see how this toxin impacts them. And all of that kind of started with this project and looking at how it affected fruit flies.
Thank you. One of the things I really enjoyed about your talk is that you really walked us through exactly what that research looks like.
Thank you.
And I really appreciate that. That was fascinating to me. It's also just amazing that you're working with something that's so tiny, right?
Microscopes were involved every step of the way.
And the fact that you're measuring how much cornmeal the poor thing eats, that's got to be, what, like 2 grains of cornmeal or something per
We actually measured it
meal.
by looking at their mouth parts under a microscope and seeing how many times they extended them to intake food over the course of a few minutes. So it's definitely easier than having to measure the cornmeal before and after they fed, but it was still a little rigorous.
So Oscar, thank you. First, I want to ask, the provost asked you about the nerves and the hunger. My understanding from sepsis is that it's often referred to simply as it's a blood infection. It engages the blood. And can you give me some sense of, is that just part of the same and just extending out to the other system, or is that something different that's going on? Is that?
Blood is definitely the area where it affects the most. As this toxin enters the bloodstream, that's what kind of initiates the immune response that ends up affecting the rest of the body. The bloodstream is where most of the cells related to your immune system are, like the white blood cells. So as this toxin enters the bloodstream and from there travels to organs and nervous tissue, the immune cells in the bloodstream follow. So we are taking a look at after they've already gotten to the organs, after they've already gotten to the tissue and nerve cells, and seeing the response from there.
So I have to apologize if my demeanor, we lost our son to sepsis. So I just thank you for this research.
I'm so sorry about that. Thank you for your time, everyone.